Okay, so first I wanted to say that I'm really happy that we managed to have one last Contra meeting, and I'm happy that I could make it in the end. So as in line with all of this session, I will also talk about the DNA damage and repair, and not that much about mutations. So we can think of my presentation as more about understanding how the mutations come about. So it's going to be more like basic biology of cancer. So to give a bit of a background why we are doing the things that we are doing, what is our motivation, is, as you all know, we often study the mutations in the context of how they vary along the genome, here in red, mutations along the chromosome 14 of the human genome, in relation to genomic features, like here in green expression, and in blue, the replication timing. And this is one of the very frequent analyses that is being done many times by many different people with different features, different sets of mutations, different signatures. And you can intuitively, looking at this, see that there is some correlation. Like a lot of the times those lines go together, but there is also a lot of missing information because there is some separation in between. And this can be explained, as also Sarah was explaining, mix of different features. They are also interrelated. There is something more there, and it also works on different scales. But as already was introduced, and I will focus here specifically on UV light damage, mutations come about first from some lesions that then get repaired or not, or get repaired not properly, and we end up maybe with mutations at the end. So in the case of UV light, what it induces most frequently is it links two pyrimidines together, forming this kind of bulky lesions that we call dipyrimidine photoproducts. And two most frequent ones are the CPDs and 6,4-PPs. They are just linking the pyrimidines in a slightly different manner. Then the main repair mechanism, as Sarah also already mentioned, is nucleotide excision repair. That is this big complex of proteins that basically comes, sees the lesion, cuts out the DNA with some additional bases around it, and then it gets filled in later. And then, if we are unlucky and this whole process wasn't successful, we end up with what we all know as mutational signature 7 mutations, which are characterized by C to T, but actually because we have the dipyrimidines, it's CC to TT that is behind it. So actually, the damage and repair, as you have seen, are also studied in the context of the chromatin features. Like, for example, here from our lab, an explanation on how the feature of nucleosomes and linkers, so having the DNA that is either wrapped around the nucleosome or is between the two nucleosomes, how it relates to the distribution of damage, excision repair, and mutations for the UV light specifically. So you can see that here, the UV damage is more or less flat in the context of this feature, while excision repair is active in the linker regions. And these two things put together give us the pattern of mutations that we see in the end, where we have more mutations in the nucleosome-bound DNA, right? So that's why it's important to understand how the damage and repair come together and how this relates to what we see at the mutation level. And this is how this is usually done. Usually we take a specific feature or a set of features, we partition the genome according to it, and then we look at the DNA damage and repair and mutations in that context. What we do in my project instead is we want to put away all of the genomic features away for a minute, and we take only the DNA damage and repair, and we try to partition the genome according to the exactly dynamics of DNA repair alone, and only we come back to the whole genomic features later to understand how this partitioning relates to that. But for now, we put all of the genomic features away. Let's focus on DNA repair in the context of UV photoproducts. And this is the team that has been involved in the project so far. So I will very shortly go through the setting because it's complicated, but I will be short. So we have actually different efforts that are trying to map these lesions, as also Pepe has mentioned. One of those is UV damage maps, where they basically take a plate of fibroblasts. They induce a short UV light pulse that ends up producing the dipyrimidine photoproducts. They look both at CPDs and 6,4-PPs, and for both of them, they prioritize the two most frequent pyrimidine pairs that the lesions appear in for both of those types of damage. And then they are interested exactly in mapping this type of lesions. So I will not go through the experiment, but the idea is that they pull down fragments that they can sequence and identify with a precision to each dipyrimidine where the damage has happened and where it didn't happen along the genome. So then, if we think about looking at the whole genome scale, and we want to count the amount of damage that we have from this kind of a damage mapping experiment, let's say in 100 kilobase chunks, we get this kind of a picture of some distribution of damage along the genome. What those experiments give us more is that they actually do this sampling at different times after the exposure to UV light. So for example, right after the pulse, so 0 hours, 1 hour, 2 hours, 24 hours. And we get then this kind of different distributions of damage at different time points after the exposure. And we know what is responsible for the changes in this landscape. It's the DNA repair, right? So, okay. So what we can actually utilize this to understand also the DNA repair. And this was already proposed by the original authors of the method to subtract one time point from another. We go a bit further because of the complexity of the data. We have to do a lot of normalizations. We modify the method slightly, and we compare each two consecutive time points. So each time interval, we compare the two damage distributions to obtain the divergences, which we treat as this kind of a repair proxy to understand more or less the amount of repair that has happened in that time interval. Yes.
So I'm not sure I understand everything of what you guys are saying. So Sarah was talking about the two strands and that there's a difference. And you are showing in these other plots that were visible, so you have a distribution. And I assume that the distribution across the genome is a summary
of both strands, yes.
Would it make a difference if you would have analyzed it on the two separate strands? So would you? So
because of us looking in the way that we are looking at this, and as we are looking in very big chunks as well, and how this UV damage specifically looks like, we analyze things with taking the two strands together. Because we are looking at really big chunks of the genome. In our case, it's 100 kilobases. We go down to 10 kilobases. And we see that it's really, we don't have the resolution to go to look at the specific strands in the context of this analysis. The experiment itself provides you with the information that you can identify which strand the damage happened. But just for the sake of this analysis, it doesn't really make sense for us to look at the strands separately. Okay. So basically, we do this subtractive damage seek in a bit of a modified version, and we obtain proxies of repair from the damage. But actually, the same lab has generated also repair maps. So what they did is they treated the cells in the same way. They have damaged the DNA. And what they do is then they know that the nucleotide excision repair comes at some point, and they try to map the excised fragments that it has taken out from the damaged DNA. So again, I will not go into depth, but they try to, again, pull down fragments, also in some way trying to take into account taking the fragments from the excision machinery itself, and then they get places of the dipyrimidines that most probably were being excised by the machinery. So if we have also the repair maps, why don't we use just them? So as the authors say themselves, I will go shortly. I don't know why it's moving without me touching, but very sorry about that for some reason. So XR-seq is way more sensitive in general than the subtractive damage seq when it comes to looking at specific regions of DNA and judging the amount of damage that is happening there. So in theory, we should use XR-seq, right? But the problem is that those excised fragments have a short lifetime before they get destroyed. So the XR-seq only reflects around 10 minutes of the excision process from the sampling time. And we are not sampling every 10 minutes. They are sampling every some hours, right? So this means that we have those snapshots that are very accurate, but we lose all the information in between. And so we have actually tested running our models that I will describe in a moment in separating the data of the subtractive damage-seq and the XR-seq, putting them together. And we see that it's actually very important to analyze both of them together because they give us complementary information. So, okay, we take both types of data, we count the amount of repair happening, and we preprocess the data a bit to have some encoding of the repair happening in different 100 kilobase chunks. For the damage-seq, we have the encoding for each time interval, each damage type, and each dipyrimidine. For the XR-seq, we have only time points and the damage types. They don't look at the dipyrimidines. So what do we do now? How can we now recover the repair activity, group the chunks of the genome, and understand whether there are any specific dynamics in different parts of the genome that we can uncover from this encoding? And the way that we approach this is we use hidden Markov models that are very frequently used for this biological sequence data like chromHMM that was mentioned yesterday to do that. So like chromHMM infers the chromatin activity states. We are trying to use similar methods to try to infer repair activity states. You can think about it this way. And it's because we need methods that will take the chunks with the encoded repair and try to cluster them based on this trajectory of the damage and repair through time. And that will also, in borderline cases, make the chunk inherit the cluster from their neighbors. And we use for this specifically sticky HDP-HMM, sticky because there is the persistence of states. So this method automatically tries to infer a very parsimonious set of states. And the HDP comes from the hierarchical Dirichlet process that is used to infer this most finite number of states, the emission probabilities and the transition probabilities. And then we use the Viterbi algorithm to allocate the states after this process. So if we look just at chromosome 1 and 100 kilobase chunks and the assigned states as a result of this whole process, this is what it would look like. So we can see that there is some evolution of the states along the chromosomes. As you can see, there is a lot. How we handle it, I will get to it in a second. So how, yeah?
Can I just ask?
Yes.
You said you limit the number of states, right?
Yes.
But is it the usual kind of hidden Markov models that we typically have in bioinformatics where you have stages, and in principle you have the same set of states at each stage? So when you go along the genome, will you be able to visit the same state multiple times?
Yes.
Yeah.
Yes. That's exactly the idea, yes. So as you can see, even this very dark blue state, that is, I think, state 0. Well, 0 is not a good example, but it reappears multiple times. And it will reappear also through the different chromosomes. So we think that 40 is not manageable, but we try to take kind of a similar approach to the chromHMM in a sense that we are going to then soft merge the states. So group states in some way into families that we can name in a general way, and then we can characterize within each family, each state a bit more in detail, but I'm getting to that. So now, how does the state relate to the data that we have had? Very shortly, the way that we try to represent it so that you can understand what we are actually finding. This is one of the states that we have found. And the data that comes in, you can see here is the 6,4-PPs, the dipyrimidine TT, and all the different time points that we have had for this damage. Then we have the 6,4-PP, the other dipyrimidine, the CPDs, and on the right we have the repair mapping for the 6,4-PPs and the CPD. And this is relative damage that is left. So this kind of scores are trying to indicate how much, how much damage we have as we go through the time points. And on the right, we have the relative repair activity. We say relative because of how we normalize things within the time point. You can see that it looks like at 48 hours, for example, for the CPDs, it looks like we have more damage. We have relatively more damage in that time point in this state compared to others because of how we normalize the data. So this is supposed to indicate that in this state, we have more damage than in the others at the end, which means that less damage was repaired compared to other more efficient states. And then we can see that it's also reflected in the XR-seq that is very low at the beginning for the CPDs and only starts getting activated towards the 16, 24, and 48 hours. So we can see that we can actually uncover some dynamics when we look at the states related with the repair activity. So the approach seems to have worked. Then, as I probably don't have much time, I will just say shortly, we cluster the states to obtain those families that I have mentioned that are now encoded on the top with the color coding. And we do it exactly based on this kind of a representation that I have shown you in the previous slide. We encode it in some way, we cluster based on that, and that's what the families that we obtain. And then now if we look at those same logos that I have just shown you, so those dynamics, but this time only for the families, instead of looking for the 40 states, we're looking only at the families, getting the artifacts away from the data, we can see that there is actually some spectrum of the repair activity. So very easily, if we compare this state, for example, and we have relatively less damage at the end to the first state that has relatively more damage at the end, we can intuitively understand that the dynamics change from slow, later happening, less efficient repair to earlier, faster, and more efficient repair as we move through the families like this. And what we also see, but I will not get into the details, is that there is differences for different types of damage. XR-seq also sometimes behaves slightly differently than what we see from the subtractive damage-seq data. But most importantly, we are recovering different repair activities. So as I said, we put away all the genomic features for some time, right? We only look at the repair data. We have uncovered some spectrum of repair dynamics. And now we would like to understand if those dynamics in any way relate to genomic features. So here I just put a selection of few, but we have looked at many. And if you have any specific one that you would like to understand, you can let me know. But to summarize this for you, as we have here the spectrum of dynamics of the families of states on top, that it goes this way towards the more early and efficient one on the right. Here, what is this blue thing is a lot of depletion in the active marks, so things related with genic regions, expression, etc., while there is some enrichment in things like lamina-associating domains or repressive marks. And we see it as we look through the different features. Another important thing to notice is that apart from there being this, on the left, slower states related with the less active parts of the genome, and the ones on the right that are faster related with more active parts of the genome, we can see that there are some differences between states. Sometimes it's just one feature that we see differs between, makes the states different. So it might make sense to, apart from keeping the families, to keep very specific states as well and understand each of them and why the model actually uncovered slightly different dynamics and if it happens to correlate with some feature in particular.
1 more minute.
Okay, I'm getting done. So we also looked at replication timing. So just to let you know, in our plot, it goes the other way than in the plot on the right. And if we again plot this dynamics of repair, we can see that the less efficient repair happens mostly in late replicating regions, while the early replicating regions are characterized by more efficient repair. So to summarize, this is still work that is not finished. We're working on the manuscript, and we know that we need to characterize everything in way more depth. But what we already see, that there is this spectrum of repair states, as I said, from later, slower, and less efficient repair in regions that are mostly repressed, packed, inaccessible, close to lamina, or late replicating, to going through the spectrum of repair states. At the same time, we are uncovering this continuum of epigenetic states, epigenetic features, sorry, and the more efficient repair happens in genic, expressed, transcribed, accessible, etc. regions. So to summarize, what we think we are bringing with this is a new framework for studying repair and understanding mutation rate variability. Our framework happens to infer different repair activity states successfully. And what we think is very important is that just based on this repair dynamics, just based on the DNA repair data, we can actually uncover some features of the cell genome and epigenome from that. we showed this proof of concept for the UV data, but in principle, and that's our plan for the future, is to apply it for different types of DNA damage and repair, including some things we are doing at our own lab, like, for example, studying the chemotherapeutics-induced damage as well. And with this, I would like to thank everyone here and my lab and the funding, and I will happily take questions. Yes, Magda?
So just judging by the eye, I'm not really convinced that those patterns look like faster or slower damage and repair. So what method have you used to compare that?
So we know that we have to work on the representation, but if we just even overlay the different dynamics on top of each other, it becomes very, very clear that the dynamics are different. Also, if we represent the logos in a different way, if I go now, this is also another representation of the logos, but this just, why is it switching? This just takes only means into account of those mini scatter plots. And you can still see that here we end up having more damage than here, right? Like even eyeballing this, it's very easy to see that there are differences, right? And then what we have done also was just overlaying the different logos. So we call this on top of each other, and you can see the differences in the patterns that are very, very clear. Yeah, we still have to work on the representation, but yes.
All right. Thanks a lot, Hanna.
Okay, thanks.
And we now have 20 minutes break.